Hi everyone, this is Mrs. Ortega here. I am starting the first video of our online teaching. So we left off in plants. I can believe it or not, that was about 2 weeks ago where we were discussing plants in chapter 30. And if I am not mistaken, we left off here with angiosperms. So that's where we're going to pick up today. But before I get going on our PowerPoint lectures, I just wanted to give you an overview of the next few chapters before our exam, which is next week. I know it quickly approaches, and unfortunately, this is just the best way to do things. So what we're going to do is look very briefly at this chapter, as we covered most of it already. Then we're going to look at chapters 35 and 36. And possibly those in separate videos, or at least 36 will probably be in a separate video. That way I can maintain the order of these a little bit better. So for chapter 30, we just have a very little bit to go over. Then we'll look at 35, 36, which I've narrowed down some of the material. Hopefully a lot of it is review from lab class, and so we will hopefully get these in all together nicely for you to study for in the next week. So picking up in chapter 30, we were looking at seed plants, the gymnosperms and the angiosperms. The gymnosperms are the cone-bearing plants. We have 4 phylum. Those are listed here, as well as in your lab. Let's see. Here we go. They are the Cycadophyta, the Cycad group, Ginkgophyta, the Ginkgo biloba, Gnatophyta, and Coniferophyta. So all those are known for having cones. And so we had gone over all that and looked at angiosperms, which are the flower and fruit-bearing plants. And I believe we had covered all the way up until right about here. Maybe right here or right here. This slide is not one that you need to know, so don't worry about that. And so we're going to start here, and hopefully that's where we left off with the angiosperms. If there is something I'm missing, please let me know, but I believe this is where we were. So angiosperms are the flower and fruit-bearing plants, and they're divided into 4 groups as well. These groups, you need to know the names of those, and so we'll take a look. But initially, they're divided into 2 groups, and those groups are the monocots and dicots. After rearranging of them, now they're monocots, eudicots, basal angiosperms, and magnolids. That wasn't always the case, though. And so the 2 main groups are the monocots and eudicots. The other 2 groups are very small and have very few species. So keeping with the main characteristics of the monocots and eudicots will be something we want to know for the angiosperms. So this page has a very nice orderly diagram that shows the differences, so the main differences between monocots and eudicots. So for their seeds, inside their seeds, you have the embryo that develops, and the first true leaves are called cotyledons. Those cotyledons are either in one formation, so 1 cotyledon, or 2 cotyledons based on monocot or eudicot. So monocots, given its name, has 1 cotyledon, 1 true leaf growing on that embryo in the seed, whereas some eudicots have 2. And they have also other physical traits that you can see as well, like leaf veins. The leaf veins and monocots are usually parallel with the veins within the leaf are parallel to the midrib or the central vein in the leaf, whereas eudicots, they're usually net-like or branching. Monocot stems have scattered vascular tissue. You might have seen the stem models in lab. The monocots, the bundles, the vascular bundles are all scattered throughout, whereas eudicots are arranged nicely in a ring. Looking at monocot roots, they're usually fibrous roots, like small networking fibers, compared to dicots, which are a large central root in the middle, the taproot, that has smaller roots branching off of it that allows for deeper anchoring and more stabilizing structures. For pollen, they also have differences in their pollen grains with openings, or those openings that allow for the sperm to leave the pollen grain and then go fertilize the ovule, or the egg and the ovule. Then flowers, you have the organs or petals, are usually arranged in multiples of 3 for monocot, and for eudicot, it's in multiples of 4 or 5. So those are some main characteristic distinguishes between the monocot and eudicots. And so in terms of angiosperm classification, you just want to know the 4 groups. The basal angiosperms are shown here. There are 3 small lineages that make these up. They include water lilies, star anise, might be some you're familiar with. But really understanding just the name here. Magnolids include the magnolia group, which is very popular in the southern states. They also include laurels, black pepper plants, and they are more closely related to the monocots and eudicots than the basal angiosperms. So if you're looking at a phylogenetic tree, tree would have basal angiosperm first. I'm going to just abbreviate that BA, and then the magnolids, and then the monocots and the eudicots. And so that order itself is not something you necessarily need to know, but it might help in understanding. The basal angiosperms are like the basal taxon, or the outgroup, and then everything else falls below that. All right, the monocots here, more than 1/4 of them of the angiosperms are monocots, and it's a very large group with many different examples. Shown here are the orchids, grass, and palm trees. And then the eudicots, we have more than 2/3 of the angiosperms are eudicot. So if you had to pick the biggest group, it would be the eudicots. And we have the snow pea and dog rose examples shown here, along with the Pyrenean oak. But there are very, very many examples. Right, and then the last slide here is not something we need to focus on, and knowing that you're probably overwhelmed already, I'm going to try as we go through all these things to let you know what to focus on. And so this is a very nice section where I like to usually go through each of these different compounds that are made from plants, specifically gymnosperms or angiosperms. And so some of these are still in use, and some of them are largely not used anymore in terms of medication. But it is here if you want to take a look. It's great interesting to look at. It's just none of this will be on the exam, so that's why I'm not going to spend more time looking at them. And so that's why we are going to have a very short part for chapter 30. I'm going to combine that with 35 in one video for that reason. Obviously, there are lots of threats to plant diversity, but again, that's not something we'll focus on. This is a nice summary page here of the concepts we discussed the last time. So if you want to take a look at that, I would suggest doing so. All right, so that's it for chapter 30. Chapter 35, here we come. So this chapter covers plant structure, growth and development. This should correspond pretty closely with your chapter 25 in lab, plant root stems and leaves. So I know you will be looking at that updated review guide. And on the review, chapters 29 and 30 are pretty short, whereas 35 and 36 are kind of long. And that's because 35 and 36, I want to be very specific with what you need to know. And so those are much more specific. If it's not on there, then don't worry about it. And as we go through, I will do my best to make sure we cover everything that you need to know. All right, so plants, they have organs and tissues and cells, just like we have organs and tissues and cells, but their structure looks a lot different than ours. And so you do want to understand an organ is several types of tissues that work together to carry out particular function. And a tissue is a group of cells consisting of one or more cell types that work together for a specific or specialized function. And so for plants, they have three basic organs: roots, stems, and leaves. And so these are organized into systems, just like our organs, like our heart. Our muscles are organized into systems like the circulatory system, the respiratory system. They have a root system and a shoot system. So the root system is going to take in water and minerals from the soil, from the ground, and send it to the shoot system, which consists of stems and leaves. And so the roots rely on sugar made from the shoot system in order to produce energy for life, so they can take in water and minerals. Whereas the shoots, the stems and the leaves rely on the water and minerals absorbed by the root system, and in turn they produce sugar through photosynthesis and send it downwards to the roots. So they have two major systems, but three organs: roots, stems, and leaves. And so everything in terms of structure shown here: the shoot system, the root system, they have a taproot example. And then with leaf structure, they have the blade is a leaf tissue, the mesophyll tissue you might be familiar with, that has all the green chloroplasts, usually about 30 to 40 chloroplasts per cell there. The petiole is the stem-like region that connects the leaf and its veins to the stem. So the petiole connects the stem to the leaf, and it's part of the leaf. In terms of the stem, you have buds on the stem. So the bud on the very end here, this one right here, that's the apical bud. That's going to be the very end of the stem. That will make it taller, by growing upwards and making more leaves and more stem. It also needs to have cells that are still dividing to make more, right? And so that could be here as well as the end of a branch too. And so those are apical buds, whereas the stem has also buds on the side. These are axillary buds. They can form new leaves or new branches. And then in between, on the stem where there's nothing there is called an internode. Any region called a node is a branching point for either a petiole, for a branch, or for a reproductive structure like the flower. All right, so let's take a look at the first organ, the roots. Okay, I'm checking here. So root is the organ with important functions. They're shown here in anchoring the plant, absorbing minerals and water and storing carbohydrates. These here, sorry, still working on my computer skills. All right, and then the primary root is called the primary root because it's the first root to emerge from a growing seed, and it forms branches to make lateral roots that will help with anchoring and water absorption for the plant. So the primary root's the first one to emerge, and usually from this the taproot develops. This is going to be the centralized root that all branches come off of, and is going to help with anchoring the plant, preventing it from toppling over. And most tall plants will have a taproot. And so you want to know what a primary root is and how that compares to the taproot. Right, for fibrous roots, they have a bunch of small networking roots that are not connected by a large central root. So they have lateral roots that arise from what we call adventitious roots, which is right here. Cover it just a little bit. And the root hairs is something else here as well. They are near the root tip and increase the surface area of the root. And so here in this example picture, the root hairs are right there on each side. So root hairs just increase the surface area to absorb more water and minerals. They are tiny little projections that usually get destroyed when you transplant a plant, when you dig up a plant from the ground and put it somewhere else. All right, and so there are specialized roots or new adaptations for roots, and all of these are shown here. These are great examples of how roots are not always that simple, but we won't need to know them, so you can go ahead and don't worry about that one here. Listed on our slides, not in this section yet, but we will get to it, I'm assuming. Let's see here. Yes, we'll get to it with roots. There's also 4 zones, and since we're here, I might as well. For roots, this may be something you saw in lab, that there are 4 zones at the end of a root. The first zone is the root cap, which is right here. So that's one. The root cap is going to be a collection of usually dead cells that will prevent the root from getting destroyed as it digs deeper into the ground. So it's you can think the hard end of a shovel. It's going to be strong and sturdy, so it's going to protect the growing root that's digging deeper. Then you have the zone of cell division, which is right here. So this is 2. This zone is going to be the zone where cells are dividing and making more cells. And so that is adding more cells, elongating the root. Which brings us to the next section here, the zone of elongation. This is referring to the cells themselves are getting bigger. As they get bigger and get larger, they're going to what we call elongate. By their cellulose fibers are going to stretch in one direction, which happens to be for the root up and down, so it's going to make the root longer. And so the root elongates, and that's because those cellulose fibers of the cell, fibers of the cell wall of those cells are elongating as they're getting bigger. So cells are getting bigger there. And then lastly, zone 4, which is zone of differentiation, which is right here. This is where cells will become mature and they're going to specialize in their function, which is also called differentiation or maturation. And so here you're going to have cells that are forming root hairs, that are specializing in their vascular cylinder transport or functioning as storage in the for storing sugar in the cortex. There's different specializations that can occur there. So those are 4 zones of the root, and I believe that is everything for the root. So understanding their primary functions, the primary root, taproot, fibrous roots, root hairs, and then these zones here. Going back to our next section, stems. So stems is the second organ we're going to look at. Stems consist of nodes and internodes. So nodes are any points at which leaves are attached, branching occurs, or reproductive structures like the flowers come from. Any region in between those points are called internodes. And then on stems they have buds. So as we mentioned, the apical bud is going to elongate the shoot. The axillary bud has potential to form a branch, a flower, another leaf, or thorn. And they also, like roots, have modified stems, are on the next slide. So these are shown here, and you'll want to know these, so keep this slide handy. So the first one, rhizomes, they are horizontal shoots, so they're actually stems that are below the surface. So this is going to be seen in many different plants, but it's, the structure itself is actually more of a stem than a root. Even though it looks like a root, it's technically a stem. So that's a rhizome, is a horizontal shoot below the surface. Whereas a stolon is going to be a shoot or stem above the surface. And so the example here is the nice strawberry stolons that run horizontally with the ground above the ground and make more strawberry plants there. And then lastly, tubers. These are enlarged ends of shoots for food storage, usually below ground. And so the potato shown here is actually a stem that's enlarged, and so that is functioning as storing sugar. So you want to know those 3. Right, on to the third organ here, the leaves. So leaf is functioning in photosynthesis, where they will take in light, exchange gas, dissipate heat. Some leaves functioning in defending the plant from pathogens or herbivores. And so for leaves, you want to know the structure of the blade and the petiole. The blade is the flattened region of mesophyll cells, and then the stalk that connects the leaf blade to the stem is called the petiole. And then their vein structure is noted here. But that is not something that's on here, so you can just ignore the veins for this portion of the review. Alright, but for monocots and dicots, you do want to know that from the last chapter. So let's see. Simple and compounds also a large division between leaves in terms of classification, but we don't need to study that here. Specialized leaves are also shown, so these are unique in function tendrils like to anchor plants. Spines on the cactus are actually the leaves. The green portion that you are familiar with is actually stem. Reproductive leaves growing on the end of the succulent leaf is also a unique function. And then storage leaves and that red onion there. The stem is actually part of what we normally think or consider as the root. All right. So tissue systems. So we've looked at the 3 organs. Now let's look at tissue systems for plants. So overall, you can see these are color-coded. We'll look at the dermal tissue system, which is here in blue. That's equivalent to our skin. It's going to be outermost covering, and it's going to function, protection, support, other things. Then you have your vascular tissue in purple. This is the transport tissue of water, minerals, and sugar. That's kind of like our circulatory system. With our blood system, transferring nutrients and molecules to all parts of the body. Then you have the ground tissue, which is in yellow here. And that's your kind of body filler tissue. That's like our fat cells, our connective tissue. It's going to kind of hold everything in place. So let's take a look at these in terms of their systems and components. For dermal tissue, we have the epidermis, periderm, and cuticle that are listed on our review. So let's take a look at those. So the outermost covering is a waxy coat, and that's called the cuticle or cutin, is what I've seen other. Okay, let me try that again. Down here, cutin. Is also another word for the cuticle that helps, is that waxy coat that prevents the water loss from the plant. Then you have the, in non-woody plants, then you have the epidermis. That's going to be the first like skin layer. It's going to be the usually on the leaf, that's like your upper and lower epidermis. That might be something you're familiar with. In woody plants, the epidermis gets replaced by periderm. And so that's what you'll want to know. So for non-woody, or for both plants, the first layer is the cuticle. Then for non-woody plants, it's the epidermis. And then for woody plants, it'll be the periderm. So I think we have a slide here. This will be a non-woody plant because the stem is green. If it's woody, the stem is going to be brown. And so that makes you know that the cuticle is the first layer here, then which is right there, then the epidermal layer is right there. Oh. So this is the epidermis. And so if this was a woody plant, then the epidermis would become the periderm. So that's the extent of what you'll want to know for that little section there. In terms of vascular tissue, we'll see this multiple times if we haven't already. The vascular tissue system is made up of xylem and phloem. Xylem conducts water and minerals from the roots of the plant to the shoots, and the phloem transports sugar from the leaves and the stems to the roots. And so that is a huge component of this chapter, and we'll see it multiple times. So that's why it's not necessarily listed to know this particular slide, but we'll see it in multiple slides. So in terms of ground tissue, the pith and the cortex are things you want to know. So anything that's internal to vascular tissue, that means it's more central or towards the inside of the tissue will be the pith, whereas if it's outside of the vascular tissue, it's called the cortex. And so that's just to help define where it is according to vascular tissue. All right, so that's to the extent of what we'll want to know there for those things. Now we'll move on to cell types. So cell types are plants. Hopefully you've seen, but if not, we will go through these and what to know from them. And so for lecture, I like to focus on the functioning, as hopefully you're learning their structure in lab. So keep that in mind. So the first type are the parenchyma cells. When parenchyma cells are mature, they have very thin, flexible walls and are least specialized. And they perform the most amount of metabolic functions and do retain the ability to divide and differentiate. So in terms of functioning, a lot of cells in the cortex and even the pith will be parenchyma cells. And so these are cells that are going to be performing a lot of different roles for the plant. And it helps that they aren't specialized for just one function. They can perform a lot of functions, and they can divide and form new cells, form new types of cells even. And so mature parenchyma cells are kind of the normal functioning cells that you need and that the plant needs. And compare that to the collenchyma cell. Collenchyma cells are known for their uneven cell walls, but for us, we want to know that they provide support without restraining growth. And so this is a key function here, in that they are not able to do all the things that parenchyma cells do, but they do provide a flexible growth by having sturdier cell structures. They're providing support, but they don't inhibit or restrain the plant from growing more, and that they are alive. Some of the cells we'll see are dead, and so when they're dead, they are not flexible whatsoever. So if the plant tries to grow and divide around it, it's just not going to move. So collenchyma cells are important, that they do provide flexible support. And a great example of these cells are found in celery. The little celery strings, if you were to break a celery stem in half, you will see these strings coming out the ends. Those are made up of collenchyma cells, the celery strings. The next type are the sclerenchyma cells. They are rigid and they are actually dead at maturity. And so this is good actually for these cells, because they will provide lots of support. They are found usually around either the stems or around vascular bundles. By having a rigid dead cell, it's going to help with support. Those cellulose fibers that stay behind are very strong, along with lignin, which is an indigestible strengthening polymer, which is right there, lignin. And so those are shown here. They're further divided based on their shape. So the scleroid cells in this pear sample are thinner and fatter, whereas the fiber cells are thinner, the scleroid cells are thicker and more irregular. The fiber are thinner and more slender. And so that's not something you want to know here, but just in terms of there's a little bit more information. For us, you want to know they are dead at functional maturity. They are going to be providing support and strengthening the plant. let's see. Then you have the xylem and the phloem. So that's why earlier in the vascular section, like just nothing from this slide. That's because we have it here. For the xylem, this is transporting water and minerals from the roots to the shoots. And they have two types of cells, the tracheids and vessel elements. So these both are dead at functional maturity. And so that's, you can think of a empty pipe to transport water in our pipe system throughout a house or a building. That's why these cells are dead. So they are hollow. They're going to be there just for support structure to transport the water and minerals quickly through the plant. So this being a dead allows for it to be hollow, and that allows for it to have faster transport. So here we have the vessel elements and the tracheids. So if you notice on this top image here, the tracheids are smaller in diameter. They're longer and slender, whereas the vessel elements are much fatter in diameter and usually a little bit shorter. And so in the connections between vessel elements, they have something called the perforation plate. It's kind of like a metal grate or ventilation grate, if you will. And so it has long, slender openings in between to allow for fast travel. For phloem, the two structures are the sieve tube elements and companion cells. So sieve tube elements are actually alive at functional maturity, comparing to the xylem here. But they are going to lack a lot of structures in their cell, and so they're going to need their companion cell to make those for it. And so this companion cell is attached to the sieve tube element and provides proteins and enzymes and other molecules needed by the sieve tube element cell. And so that's shown here. The sieve tube element is the long, hollow cell structure here, and then it has a sieve plate that connects from one sieve tube element to another. So right here is a sieve tube, another sieve tube element, and then the plate connecting them is more like a shower head grate with small circular holes. That's the sieve plate. And then they have the companion cell right here and right here that provides basically everything it needs to stay alive. And so that companion cell has to produce for itself and for the sieve tube element. And this is a Bio 1 video I'm not going to bother with as it will be just using up more of our time that's already valuable. So that brings us to growth. So we've covered organs, tissues, and cells. Now we're going to look at how these things grow. And so that will be the next portion here, which some plants have a certain size they can reach. It's just determinate growth. Others do not have a definite size that's that they reach and then stop growing. So that's called indeterminate growth. But both of these things are not in our review, so you don't need to worry about it. But in terms of growth, we have two main divisions we want to know. The first one is primary growth. This will be on the apical meristems that elongate shoots and roots. So that's making the plant taller and its roots grow deeper into the ground. Whereas secondary growth is going to make the stem thicker. And so this will be what we see adds thickness to woody plants. And so these things are done through special meristem tissue that adds more cell layers. So the vascular cambium is going to add vascular tissue. So this is going to be both xylem and phloem that get added. And then the cork cambium will add more bark tissue, essentially. We'll look at that. So these two things contribute to the secondary growth. Let's see here. All the details noted here are not something you need to focus on. So primary growth, growth in length is shown here, and that there's a cell division, forms a new daughter cell, then that cell divides and grows and differentiates. And so as it gets elongates, as it gets longer, there are regions of mature tissue. There are division, what we call primary meristem region, and then the apical part, which is, so over time, all these regions stay, they just get longer. So obviously as more apical meristem is added, more primary meristem converts into mature tissue. And so as time goes on, it continues to get longer while maintaining all three states. Whereas the secondary growth is going to be the vascular and cork cambium that will add more cells that will make it thicker. And so first the cells divide and they form, from this example, from 3 cells to 6 cells, so they divide. And those cells are very small, so after time of growth, that will make the skin, make them thicker, and that will make the stem thicker. And so we'll see secondary growth. The vascular cambium first adds a layer of xylem, which is noted here as xylem 1. And then it adds a layer of phloem, which is known as P1. Then it adds another layer of xylem tissue, xylem 2. And then it adds another layer of phloem tissue, phloem 2. So remember, xylem's transporting water and minerals. This can be more towards the internal portion of the stem, and phloem is actually going to contribute to the bark. And so that's towards the outer portion of the stem. And then the cork cambium, this is going to be more external, so it's going to be outside of the vascular cambium, and that's going to be adding cork layers that will form bark. And so that's shown here. In primary growth, it's growing longer. You have the cambium, vascular and cork cambium cells are there, but they don't start dividing until secondary growth. And so let's see, in this you have the oldest xylem cells going to be towards the center, as it's farthest away. The most recent will be towards the vascular cambium, and then the youngest will be, again, closest to the vascular cambium of the phloem, and the oldest is going to be farthest away this way. And so then you have cork outside of that, adding more cork layers to form part of bark. And so here shows you an example of growth for a stem. So this starts out obviously from the bottom as it grows from bottom to top. So this is the oldest region, which is 3 years old, and then there's a scarring here that allows you to know that the next growth is 2 years old, and then from there this gets added. This axillary bud starts growing a branch, and so that's actually 1 year old that corresponds with this as well, which is 1 year old. So by studying branches, their nodes, everything, you can, along with looking at the secondary growth, you can kind of estimate some ages of plant stems that way. Let's see here. So as we noted, primary growth makes the roots and shoots get longer, and so we looked at the zones of the root already. And so looking next at stems, there's a difference between monocot and dicot, which is shown here. Let's see. This is not something you need to focus on, so keep that in mind. I still don't think we need that. Okay, no. We have enough to learn, so moving to leaves. So leaves here, we'll look at some leaf tissues that we didn't look at before. And on the leaf tissue itself, the epidermal layer or the periderm, depending if it's, green or woody, herbaceous or woody, they have stomata. These are pores that allow for gas exchange of carbon dioxide and oxygen between the air and photosynthetic cells in leaf. In the next chapter, we'll see how stomata actually help in moving water through the xylem of the plant itself, the whole organism. And so each stomata is an opening, is a pore that has guard cells, or two guard cells that will regulate in its opening and closing. And so we'll see those in the next chapter too. In terms of tissue, the leaf tissue is called mesophyll. This is something you want to know. Mesophyll has the chloroplasts of about 30 to 40 of them in each mesophyll cell that are responsible for photosynthesis. There's two types of mesophyll that we'll want to look at. So those are listed here. The palisade mesophyll is usually the upper part is kind of more closely together, whereas a spongy mesophyll is a loose arrangement that allows for not only gas exchange but for water vapor to line the spongy mesophyll. And so that should be shown here. The palisade mesophyll is up here. It's the tight network of cells, and then the spongy mesophyll down here is loosely arranged. And so the spongy is all of this here, and then the palisade is all of that here. And then around the vascular tissue in the leaf, so around those veins, is what we call the bundle sheath cells. And so that is something else you want to know, the bundle sheath cells. There. I'm just checking our time. All right. I'm going to let this go a little bit longer than what I was trying to do, just so we can get the whole chapter in on one video. So looking at secondary growth is something we want to do as it is a good portion of our work here. And something hopefully you looked at in lab as well. So secondary growth allows for the thickness of the stem to get bigger. And so we will see how this can occur simultaneously with primary growth. So as it gets longer, it can also get thicker at the same time. And in that process, usually from the first to second year, the epidermis, which is green here, the green stem, gets replaced by periderm. So keeping those dermal layers in check also help. So epidermis is that green dermal layer, whereas periderm is more like bark. It's going to be the woody bark here. That's made as the stem gets older for woody stems. And so let's see here. Okay, this does help. I'll continue really quick. And that the vascular cambium will add layers of xylem and phloem with every year, and then they will also add more cork. And so that's shown here. So the first stem being the youngest portion, this has the xylem, which is in this aqua blue-green here. Then you have the red line, which is vascular cambium. And then you have the phloem, which is this dark brown red here. And then the epidermal layer. And so after the growth happens in the year's time, you have, or season's time, you have another layer of xylem, another layer of phloem, and then cork. So cork cambium layers added. This is developed from the phloem and cortex region in yellow. And so that produces the cork cells that will form the periderm. And so as it continues, every season we'll see it adds 2 layers of xylem. Most noticeably, we can see the xylem, the phloem's a little bit harder as that does become part of, along with the cork and cork cambium, that becomes part of the bark, which is also, periderm itself is cork and cork cambium, but altogether bark is periderm with phloem. So that should be shown better here. That periderm is cork cambium and cork together. Periderm with phloem, which is shown here, is also bark. And then the xylem are the rings that form that you can count. And so here, the vascular cambium will be the layer in between the xylem and the phloem. And then you have these rings of xylem. And so the thick portion here with fat, round cells, that's a happy season. That's the springtime when cells have enough water. And so that will be the first layer that gets added. And then the second layer, which is most notably shown right here, is usually during the summertime when water is scarce. And so it forms tiny, unhappy, small cells, and that is able to be noticed by that distinguishing line. And so you have springwood and summerwood, and these two together form 1 year. It also forms a line that you can count. So every line equals 1 year of growth. And so you can count the age that way. And so that essentially is kind of the understanding of secondary growth. You want to know what the vascular cambium adds, secondary xylem and phloem, cork cambium replaces the epidermis with periderm. The lenticels, that's something on the review. I will change that. You don't need to know lenticels. And what bark consists of. That's everything external to the vascular cambium, including phloem and periderm. So there's a couple more things here that we want to look at. So all of this, if it helps you, great. If it doesn't, it's kind of repetitive to what we've already said. So you don't need to focus on it if you understand the previous stuff. Here, the xylem. So secondary xylem, as more layers get added, the layers that are the oldest eventually will lose functioning. And so that becomes what we know as heartwood. As it's closest to the center, like our hearts to our bodies, this wood is usually dead. And so that can just over time begin to desiccate or degrade. And so that's how some trees are hollow in the center, yet have full functioning that are perfectly live trees. And so it's because that heartwood is dead and no longer functioning, whereas a sapwood that is transporting water and minerals or sap is still functioning. And so that allows for that tree to have, we have a distinguishing part of the xylem there. So as bark gets soft off over time, the vascular cambium keeps adding more layers of phloem, and in turn phloem adds differentiates and adds more layers of cork cambium and cork. So you might wonder, well, bark can fall off, yes, and so the phloem, starting with the cambium, will add more phloem. Phloem will add more cork cambium, and cork cambium will add more cork. So it continues to produce. Let's see. Vascular rays is something on here as well. Vascular rays are going to be can be in the xylem and the phloem. So if you see in lab, there is like a woody stem in lab that's available to look at, and that has nice example of the xylem and phloem ray. And so a ray is going to be usually parenchyma cells that span like a transverse, across all the tissues throughout the whole stem to help with transport of nutrients to all cells. And so you can think of it as smaller like capillaries that extend from the main vein in the center. These guys will help transport nutrients between different cell types, between the xylem and the phloem and the cork cambium. Vascular rays go across the whole stem. All right, let's see here. So this is a nice large tree that has a tunnel cut out. So the sequoia trees commonly found in California are a great example, like this tree, of where its hollow center, the heartwood, is no longer functioning, and so you do cut through some of the bark and living tissue to make a tunnel, but overall it can still maintain life fairly well at this large size. So this year I will update the review guide again. I'm sorry, you don't necessarily need to know that part. And so this also can be helpful if you like to organize the overall ideas of how those tissues develop from the epidermal layer and the phloem, xylem, and cambium and all that. So overall, these slides help. In addition, great. If not, you can't ignore them. So that will be it for chapter 35. So I will update that review, and then I'll make the next video for chapter 36. Until later, bye-bye for now.